how they pass down characteristics to their offspring. So we're going to look at a very simple form of reproduction called binary fission. And binary fission mostly happens in prokaryotic cells, so mostly bacteria cells. And we're going to look at the cell cycle in eukaryotic cells, which is kind of the beginning of seeing how eukaryotic cells reproduce. So let's look first at binary fission. So binary fission happens in bacteria and archaea because they both have prokaryotic cells. And it happens in a few eukaryotic organisms if they're single-celled. So if you had a prior class in biology, you might have studied an organism called amoeba or paramecium. And those organisms are one cell, is their entire body, they're single-celled, and they reproduce by binary fission. So we're going to focus on binary fission in a bacteria cell. So here's a bacteria cell. The outer part is the cell wall. Inside that is the plasma membrane. This kind of lighter peach yellow color, this is the cytoplasm. And remember that towards the center of the cell, there's an area called a nucleoid, and the nucleoid has one chromosome. And that chromosome is described as being circular, but it's really kind of a big twisted knot. So the first part of reproduction is the cell replicates its DNA. And when the cell replicates its DNA, if everything goes correctly, we have two exact similar chromosomes. Now, you might be tempted to use the word copy. We don't like to use the word copy. The reason is because when most of us think of copying something, you probably all put a piece of paper into a copy machine and made a copy of it. When it came out, you had your original sheet of paper, you had a new version of that sheet of paper. Here, what we're going to do is we're going to use our original DNA, this dark purple, as our starting point. And when we get done, we're not going to have our original DNA and a new piece of DNA. So we don't like to say copy because most people think of that not quite accurately. Now, if the DNA is replicated accurately, then the DNA in this dark purple is identical to the DNA in this light purple. Sometimes there are mistakes in replicating the DNA. Does anybody know what one of these mistakes is called? Well, when you have a mistake in replicating the DNA, this is called a mutation. And this is actually one of the main things that causes living organisms to evolve. Now, after the DNA is replicated, and notice we can kind of see there's a little loop that attaches this chromosome to the plasma membrane, the cell grows a little bit. And as it grows in the space between the two chromosomes. And as it grows, the plasma membrane and the cell wall stretch. And since this little loop connects the chromosome to the plasma membrane, when the plasma membrane stretches in this middle area, this pushes the two chromosomes apart. Then a cross wall, some new cell wall material, remember prokaryotic cells have a cell wall, a new cross wall and plasma membrane form in between the two chromosomes. We say that the cell membrane and the cell wall are invaginating. And invaginating means they pinch in. Think of one of those long balloons if you pinch in the middle. Eventually, this cross wall and the plasma membrane completely separate our original cell into two cells. And each of the cells has one of the chromosomes. Now notice that these two cells that we have here are a little bit smaller than the cell that we had up here. And that's because before it gets ready to reproduce, cells are going to grow. So this cell is going to have to grow until it gets to about the same size as this one before it can be divided again by binary fission. And the same thing for this. Then, depending on the bacteria, sometimes the cells stay stuck together, sometimes they separate. And the two cells that we make are always called daughter cells. There are no son cells. Sometimes the cell that made them is called the parent cell. Now, notice that we said if replication happened correctly, these two chromosomes have exactly the same DNA. So when we look at binary fission is considered to be a form of asexual cell division. A at the beginning of a word like asexual means not. We'll see later sexual cell division changes the DNA of the daughter cells. But in asexual cell division, if we look at this original dark purple DNA, the genetic components of the chromosome of the parent cell are exactly the same as the genetics of both daughter cells. So in binary fission, the cell that starts the process and the two cells that end the process are all exactly the same genetically. So that's why this is asexual cell reproduction, because the DNA doesn't change. So when we're talking about cell division, we want to keep in mind that whether we talk about cell division being sexual or asexual, doesn't refer to what you might think of when you hear sex. It refers to whether the DNA of the daughter cells is the same as each other and the parent cells, then it's asexual. If they're not, then it's sexual. Now, keep in mind that our bacteria cells only have one chromosome. But when we look at eukaryotic cells, it's a little more complicated. If we look at human cells, for instance, human cells have 46 chromosomes. If we take those chromosomes out of a cell and we line them up end to end, they actually stretch for about 3 feet, for about a yard. Well, your cells are much smaller than a yard. So reproducing in a eukaryotic cell is more complicated, partly because there's so much genetic information. There are so many chromosomes. The cell has to be very careful to keep the chromosomes from getting too tangled up. So when we look at eukaryotic cells go through something called the cell cycle, except for a few of them that actually divide by binary fission. So with eukaryotic cells, the first stage of the cell cycle, and notice since this is a cycle, we're going to end up back where we started. The first stage is something called G1 phase. And G1 stands for the growth phase, and it's the first growth phase. Originally, scientists couldn't figure out what was happening, and it was the first gap phase because it was a gap in our knowledge. But now we're calling it the first growth phase. It sounds more impressive. Now, a couple of things happen during G1. One of the main things that happens is cells do their normal activities. Excuse me. So if we're talking about cardiac muscle cells in your heart, they're contracting and they're pumping blood. If we're talking about cells in the retina of your eye, they're detecting light and sending signals to the brain to figure out what that light means. If we talk about, nerve cells in your spinal cord, they're communicating with other nerve cells. So different cells do different things, but whatever the normal activities for a cell are, this happens during G1. Now, some cells basically are going to stay in G1 for long periods of time. And if they stay in G1 for long periods of time, they actually kind of exit the cell cycle. So if you could imagine an arrow coming out here, and they go into something called G0. And G0 means all they're doing is their normal activities. Because if they're truly in G1, besides their normal activities, they're doing something else. they're growing a little bit larger. Remember what we just said with our bacteria cells. They have to kind of double their size before they can divide to make 2 daughter cells. So if the cell is really going to divide, if it's truly in G1, it's not only doing its normal activities, it's growing bigger, and it's making some proteins that are going to be involved in cell division. And one of the most important proteins is something called S-cyclin. Now notice the next phase is called the S phase. S-cyclin is a protein that, in a sense, the cell counts how much of S-cyclin it has. Keep in mind it does this counting in a chemical sense. But if the cell makes enough S-cyclin, then it moves into the S phase of the cell cycle. If it doesn't make enough S-cyclin, it stays in G1. So let's think about a cell that makes enough S-cyclin that it goes into the S phase. When it goes into the S phase, S stands for synthesis. And this is when the cell synthesizes DNA or when the cell replicates its DNA. So in your cell, you started out with 46 chromosomes. At the end of replication, temporarily, you're going to have twice as much DNA. Now in the S phase, the cell again makes some chemicals, and these chemicals cause it to go into G2, the second growth phase. And in the second growth phase, the main thing that the cell does is get ready to divide. And then the last stage is often abbreviated as M. And there are really 2 processes that happen during M. There's mitosis and there's cytokinesis. And we have to have both of these happen to have cell division. And there are 4 stages in mitosis. There's prophase, metaphase, anaphase, telophase. And mitosis is just the division of the nucleus. So this is dividing up the chromosomes. And cytokinesis, cyto means cell, and kinesis refers to division. Cytokinesis is the division of the cytoplasm and all the organelles that are in the cytoplasm. And this typically happens towards the end of mitosis. So mitosis and cytokinesis almost always go together. Now, in prophase and metaphase and anaphase and telophase, we can actually see the chromosomes in a cell. Since we can see the chromosomes, when scientists were looking at cells originally, they could see that the nucleus was obviously doing something. You could see the chromosomes. But if we go back to G1 or S or G2, you can't see any chromosomes in our cells. And because you couldn't see any chromosomes in the cells, G1 combined with S combined with G2 were originally given a name interphase. Inter means between. So remember, this is a cycle. So if we think about the end of telophase and cytokinesis, interphase is between telophase and cytokinesis and the next prophase. It turns out that even though biologists originally thought like interphase was described as a resting stage when the cell wasn't doing anything, it was just kind of sitting there goofing off. Remember what I said? This is when the cells actually do their normal activities. Now, keep in mind when we look at cells in your body, some cells, like cells in your skin, go through mitosis on a very regular, constant basis. You lose millions of skin cells every day. So in the bottom layers of your skin, there are some layers of cells that divide by mitosis. And when they divide by mitosis, they push one of the daughter cells up towards the surface of your skin to replace some of those cells that are being worn off at the surface. On the other hand, some cells in your body, like cardiac muscle cells, don't really divide by mitosis. So this is actually one of the major problems that people have when they have a heart attack. When you have a heart attack, usually some of the cardiac muscle cells in your heart die. Cardiac muscle cells are what pump the blood. So after some of your cardiac muscle cells die, the cells that are left have to work harder to make up for, those cells that are dead and aren't doing their part of the job now. So, and we have some cells that, like cells in your liver, that don't normally divide, but they can divide in an emergency. If we cut a chunk of your liver out to give somebody a liver transplant, your liver cells, as long as they're healthy, will start dividing and replace that chunk you're missing. But normally, liver cells don't die. So when we look at cells in your body, some of them divide by mitosis very frequently, some hardly ever, and some never. So let's think about what we're going to see when we look at prophase. So on the left side of this diagram, we have 2 chromosomes. And then the arrow represents the process that occurs during replication. And the right side is what we would see if we looked at these 2 chromosomes after replication. So let's talk a little bit about human chromosomes to understand this diagram. We can say that people have 46 chromosomes, but another way of thinking of the 46 chromosomes in a human cell is to think of them as being 23 pairs of chromosomes. And your chromosomes are actually numbered. There's chromosome number 1, chromosome number 2, number 3, all the way down to chromosome 22. When we look at the chromosomes, if you look at your 2 copies of chromosome number 1, so let's say that this is chromosome number 1, the blue chromosome represents the chromosome you inherited from your father. The red one represents the chromosome you inherited from your mother. So we're looking at chromosome number 1 from your father and chromosome number 1 from your mother. Now, if we look at a person, a typical chromosome has about 1,000 genes on it. And each gene is the instructions to make a protein. Now, chromosome number 1 from your mother and number 1 from your father have the same genes on them. Now, let's be very specific by what I mean when I say they have the same genes on them. Say that there's the gene for eye color on chromosome number 1. And maybe it's about halfway down this upper bulge. Then halfway down the upper bulge on chromosome number 1 that you got from your mother will be the gene for eye color. Now, keep in mind when we say the gene for eye color, we're really just saying this is where the instructions for making the protein that gives you your eye color is found. We're not saying that it's the same instructions. The gene that you inherit from your father could carry the instructions for brown eyes. The gene that you inherit from your mother could carry the instructions for blue eyes, but it's the gene for eye color. Or it could be the gene for whether you have type A blood or type O blood or type B blood. So there are, on average, about 1,000 genes per chromosome. Now the small ones have just 100 or so, and the big ones have several thousand, but yeah, that's a good average. These 2 chromosomes that have the same genes on them are called homologous chromosomes. So if we take homologous and we break that word down, homo means the same, and logos means places. And this is because the genes are actually located at specific places on the chromosomes. In fact, we actually have kind of maps of the chromosomes. And just like Google Maps, you can say, okay, what's at spot 450 on chromosome number 1? So homologous chromosomes have the same gene and it's at the same spot. The instructions could be different. Now, during replication, remember what I said. The cell is going to duplicate its DNA, and if everything happens correctly, we're going to have 2 exact copies. Well, these two exact copies of blue number 1 are represented by this kind of X or H-shaped structure that we have here. And the two copies of red number 1 are represented by these two strands. So the left strand is part of the original DNA. The right strand has part of the original. The left strand has some new, the right strand has some new. But if everything was done correctly during replication, the DNA in the left strand is exactly the same as the DNA in the right strand. And that's true for both the blue chromosome and for the red chromosome. So these two duplicates of each other are called sister chromatids. And again, get used to it, guys. When we talk about reproduction, all the terms that are used are female terms. There's really no point at which we're going to be using male terms. So there are no brother chromatids, just sister chromatids. The sister chromatids are held together by an area of DNA in the middle called the centromere. And the centromere has some proteins attached to it. And you could think of this as being kind of like a staple that connects the two sister chromatids. So they're firmly attached to each other. So tomorrow we're going to be thinking about these two sister chromatids and how they act during mitosis, because we're going to have to split these chromatids up to make an egg or a sperm. Because if this is you, if this is chromosome number 1 that you received from your father, and if this is chromosome number 1 that you received from your mother, when you make an egg cell or a sperm cell, your egg or sperm only gets one copy of chromosome number 1. You might pass down chromosome number 1 you received from your father. You might pass down chromosome number 1 you received from your mother, but you only pass down one. Well, that doesn't seem fair. You get 2 copies of number 1. Why do you only pass down 1 copy of number 1? Because the other copy comes from your partner. So you pass down 1 copy of chromosome number 1, they pass down the second copy of chromosome number 1. And again, you might have the same instructions on those 2 chromosomes. You might have different instructions. So first we're going to look at how the chromosomes are passed down. Later we'll see what happens if the instructions are different, or what happens if they're the same for that matter. So are there any questions about the cell cycle for now? And notice this is only in eukaryotic cells. You would never see a bacteria cell in G1 or S or G2 or M. Bacteria cells don't go through the cell cycle. Okay, if there are no questions.